Well, it's not. I just wondered if you had tested or checked whether or not the RNAs might themselves form a hairpin loop or a secondary structure that might stop them binding to the target RNA.
What we thought is that we analyzed the secondary structure of the small RNAs, and when we were building our scaffold and adding the sRNA, the target humanics structure, probably if they have different structures, a bigger cell will make a different proportion to the linear structure. It might affect the binding to HFK, for example, and that will result in a longer process, and that can possibly explain why the decrease in fluorescence was just seen after 24 hours. So the structure of the small RNAs can be related with the different results that we have.
Okay. And the other question I had was regarding you mentioned in your human practice that you did a two-way dialogue. And you went to see some people, elderly people in some schools and stuff. Where's the two-way element of that?
The creation of two-way dialogue was basically, we went, during our visit to the elderly, we went out and we presented our project to them, which genetic modification is not some kind of scary thing crazy scientists are doing back in the lab. It's actually something that has a really great potential. So we're kind of hoping to just, through talking, kind of like bring closer that issue and just kind of alleviate any of their concerns.
Okay, thank you.
Thank you very much.
Thanks for a nice talk. Can I just ask, if your project worked, how would you implement it? Because you say that the potential application of it is antibiotic resistance. How would you implement that in hospitals?
Also das Gewehr.
So the way we think our small RNA can be a potential tool in antibiotic resistance is that we found when doing literature research that actually many small RNAs are involved in helping bacteria avoid antibiotics. And we thought that because small RNAs can not only use to target messenger RNAs but also small RNAs itself, we could use our small RNAs so that we can insert any targeting binding site. And in that way, for example, there are many small RNAs that help that when are overexpressed will help bacteria to overcome the effects of antibiotics. So if we could potentially downregulate those small RNAs or downregulate the proteins that help to acquire antimicrobial resistance, we could synergize it with the effects of antibiotics and actually make the effects of the antibiotics more better.
Thank you.
Thanks, everyone. I think we're out of time for the questions. And so give it up one last time for King's College. And next up will be Duesseldorf.
Ja, das ist kein Problem. Können wir dann gleich Fragen stellen?
13 Zuschauer. Kunst. 16. Meine Eltern sind gerade bei Ikea. Ich glaube, die gucken das trotzdem. Cool. Frage am Kanton. Es hieß gerade, dass es Kanton gibt.
Hört sich doch an.
Ja.
Aber da stand doch gerade Mikrofon an.
Ich glaube, es ist so ein Quick-Link, so von wegen, wenn ihr hier draufklickt, dann schaltet es sich. Ah, fuck. Das groß aus viel zu hell. Das siehst du auch. Ich weiß.
Mal gucken, ob das aus einem anderen Winkel besser ist.
Der
Ja,
Ton
wenn du
ist
sonst so
da.
schräg hältst. So. Nein.
Wenn du näher dran gehst, dann müsste sich die Belichtung eigentlich automatisch anpassen, oder?
Hört ihr uns?
Naja, aber die anderen Folien sind nicht so gut. Ich habe noch seine Fans vergessen.
Wir haben echt keinen Ton.
Okay, good morning for the
ist
second
eingeschaltet.
session. And so, Team Duesseldorf, take it away. Good luck.
Welcome everyone. Thank you very much for the introduction. This is Jenny. I'm Karina, and we are part of the IT team of Düsseldorf, and would like to present our project now. Before we start, though, I have a question for you guys. Who of you woke up today in a shared hotel room? Please raise your hand. Who of you guys are sharing their hotel room with someone? Okay, a bunch of people. Interesting. Now I assume that you guys all knew your roommate, right? But just imagine you were assigned to a random hotel guest. Just imagine. It could have been a hit, but it could have also been a rather uncomfortable situation. Now, did you know that microorganisms are not that different from us? They also like to share the living space, but not with everyone. As you might know, the natural habitat of microorganisms consists of many networks and interactions that is only barely understood. Now, if you try to recreate those conditions in the lab, you do this in a so-called co-culture. We realize that there are only a few stable and synthetic systems out there, even though understanding these networks is so important for so many colleagues. For example, there are only 1% of microorganisms that can be cultivated so far. 1%. This is also due to the fact that a lot of microorganisms are not require the benefits of living in networks and cannot be cultivated by themselves. In addition to that, a lot of these networks are also living on and inside our body, for example, in our gut. A lot of illnesses actually start here, but cannot be sufficiently treated because scientists and physicians just don't know enough about them. This is why we decided to make cool cultivation more accessible and decided to create a cool culture toolbox. We established 3 different independent systems that can be combined that I'm going to introduce to you now. In each system, a new microorganism is going to be presented. In our first system, we are trying to control the population size of a very fast dividing organism in order to target the problem of overgrowth. This organism is going to be regulated by using the chrome sensing system. It is usually used in bacteria as a communication method, where signal molecules are constantly produced and diffused into the environment. At lower cell densities, no gene expression occurs. At higher cell densities, however, more signal molecules are in the environment. If a certain threshold of signal molecules is reached, promoters can be activated and genes can be transcribed and translated. But how is this of any use for our project? Now in our system, we are going to use E. coli, a fast-growing organism. We are going to exchange the regulated gene with a lysis gene. So low cell densities, nothing happens. But at higher cell densities, some cells lyse. Now before testing for lysis, we actually wanted to check the functionality of our promoter. This is why we exchanged the lysis gene with the GFP gene, so that after induction of the chrome sensing system, fluorescence should be visible. On top, you can see fluorescence microscopy pictures, and on the bottom, you can see the merge of those fluorescence microscopy pictures and DIC microscopy pictures. As you can see on the left, where our construct was tested, fluorescence is visible, so our expression system works. As expected, the negative control shows no fluorescence, in contrast to the positive control, where GFP was under the control of the putative promoter. Now, after having done that, we wanted to find out if we were actually able to control the population size of our lysis E. coli. This is why we conducted this growth curve over 1 day, where we measured the optical density at 600 nanometers. Now, as you can see in green, the optical density of our lysis E. coli starts to decrease at around 8 hours. This is a contrast to the control population in blue, as well as the E. coli in yellow that carries the GFP gene instead of the lysis gene. Now, being able to control the population size of one organism, we wanted to challenge ourselves a little bit inside of why one when we can have two, which is why we created this two-way co-culture system. This is when the baker's yeast, Duesseldorf, is going to join the E. coli. In this system, we decided to use the cross-feeding of amino acids as a communication and dependency method. From now on, we are going to use the NP2 medium as our cultivation medium. It is a derivative of BG11 and specifically designed for co-cultivation experiments. In this system, Duesseldorf is oxytrophic for lysin and is engineered to produce leucine for E. coli. E. coli, in turn, is oxytrophic for leucine and was engineered to produce lysin via a non-feedback inhibited gene. Thus, both of them need each other to survive. Here, it was most important to us that both of these organisms are able to produce sufficient amounts of amino acids. This is why we did another growth curve where we measured the colony forming human for 36 hours. And here we cultivated yeast in different media, and the cultures were plated and the colonies were later counted. The control in yellow shows the growth of yeast in a molded medium where all necessary amino acids were added. In blue, you can see the growth of E. coli in the reused medium of the lysine-producing E. coli. This means that we cultivated E. coli for 1 day, then used the supernatant for the growth of yeast. As you can see, yeast is growing in this medium even though there was no lysine added beforehand, which means that the E. coli was actually able to produce amounts so that yeast is able to grow. Now, after having done this, we wanted to challenge ourselves even more. We decided to add a third organism, which you can see on the bottom. It is Aspergillus, a photoallotroph cyanobacterium, which is a very slowly growing organism. So in this system, we decided to establish dependencies based on the secretion of essential nutrients. So in this medium, 3 different nutrients are provided in a form that is only usable by one organism. So each organism needs to convert one of these nutrients in order to provide it to all the other 2 organisms. Acetylgalus works as the carbon provider, so it is able to produce and secrete sucrose. E. coli functions as the nitrogen provider. It is able to convert melamine into ammonia. Acetylgalus was engineered to convert phosphate to phosphate in order to supply these to the other 2 organisms. Thus, all 3 are dependent on each other. Jenny is now going to continue with more details.
Thank you. So, first of all, we wanted to have a look at the system of the cyanobacteria we wanted to focus on is working so far. We obtained a strain which was published before and is commonly known as CSCB strain. Here, a short view of the metabolism is shown. We wanted to produce and secrete sucrose. This works in the strain as follows. First, after induction with osmotic stress, sucrose is being produced out of glucose and fructose. This is also shown in blue in the graph. In red, you can see, by further induction with IPGG of the system, the CSCB supporter, it gets activated and secretes sucrose and the proton to the medium. So far, so good. That's for the theory. But we also wanted to have a look on if this is also working in all labs. Therefore, we had a quantification method. We wanted to measure the sucrose in the medium. Therefore, you can see on the y-axis the amount of sucrose released into the medium. And we wanted to compare the wild type, the progenitor strain, which is not known to produce and secrete sucrose, with the CSCB strain we obtained. And here you can see the comparison. The progenitor strain was not able to produce and secrete any sucrose, while the CSCB engineered strain is able to secrete increasing amounts over the time. We take out media samples before induction, 2 days, 4 days, and 6 days after induction. Okay, we are able to secrete sucrose, and this shows us that we can use this cyanobacteria as a carbon provider for our co-culturing experiment. Next, I want you to have a look on the growth of the yeast in phosphate medium. As shown before, we wanted to engineer a yeast which is able to grow on phosphate-containing medium. Here, therefore, we have different constructs. They were under the control of different, commonly used constitutive promoters in yeast. For the strongest promoter, which is the blue one, shows PTDH3 promoter, we resulted in the highest growth. So for the next experiments, we also wanted to focus to work with this concept. Now, we talked a little about engineering and providing nutrients into the media, but there were no co-culture experiments, right? But we wanted to establish also co-culture experiments. Therefore, we want you to have a look on the experiments of the growth of S. cerevisiae first, and then later on of the cyanobacteria. Here, first of all, presented the growth of the yeast in co-culture setups. You can see on the y-axis the colony-forming units. This is just the case because we were interested just to measure living cells in the culture. So in the blue graph, you see the positive control, so the monoculture of S. cerevisiae, and you can see growth, obviously. In yellow, you can see the co-culture of S. cerevisiae and E. coli, and the amount of living cells remains to be stable for the yeast in this setup. But if you have a look on the green and the red graph, where Duesseldorf joined the co-culture, and additionally, no additional carbon source was added, so Duesseldorf was functional for the carbon providation in the system, you see a decrease in the amount of living cells of the yeast. This was for us the conclusion, okay, co-culturing is possible for us, but we have a problem because there might be not enough sucrose in our system provided by the cyanobacteria. Now, we wanted to have a look on the cyanobacteria itself. Is it growing in our co-culture, or what is the case? Here you can see the relative fluorescence units on the y-axis, just because the cyanobacteria is autofluorescent, so we could measure the growth with the fluorescence units. And you can see for all, also co-culturing setups in all colors, cyanobacteria are growing in this case. Now I want to sum up our co-culture experiment, and I'm going to present you a three-way co-culture with all three organisms in one bucket. Okay, please focus first on the left y-axis. On the left y-axis you can see the colony-forming unit, logarithmic, in blue for S. cerevisiae, and in yellow for E. coli. E. coli amount remains to be stable all over the time. But this is, as I presented before, not the case for the yeast. The amount of living yeast cells seems to be decreasing. Now have a look on the right y-axis. On the right y-axis again, the relative fluorescent units of the cyanobacteria are shown. And, as expected and shown before, they are increasing over the time. This seems to the conclusion that we are able to have a co-culture for 48 hours, but we have a decreasing amount of the yeast cells that are living, and this is not what we wanted. So we came up with a model. This model is built on step-by-step calculations. We let out a few assumptions because co-cultivation is a very complex system. We wanted to make it a little bit more accessible for us. So we wanted to have a model which enables us to allow us and to see what we can expect from our cultures. So we can play with the model with different inoculation amounts of different microorganisms in this case. And this is what you see in the end. This is what you are, or we are. This is a shared hotel room with all organisms inside, communicating with each other. And we understand their communication. This is what we wanted to create. Looks beautiful, right? But iGEM is not so beautiful. It's beautiful, but it's also hard. And this is not just web lab work. We also had a lot of troubles this year. Some of you might know this postcard. I wanted to present you now our outreach and human practices. First of all, I want to focus on the integrated human practice. As mentioned before, we have three different setups, right? They are independent. And for the first setup, we had Skype meetings with Dr. Spencer Scott. He is an expert regarding to chrome sensing and is working a lot with it. And he was able to help us designing our experiments for our co-culturing or chrome sensing experimental work. Next, for our second approach, the oxotrophy-based co-culture system, we had email Q&A sessions with Dr. Shradashitur. She's an expert in cross-feeding between amino acids and between different organisms, and she had a huge of knowledge for us. That was nice. Last but not least, for our third system, the three-way co-culture, we had a personal meeting with Dr. Daniel Duquette. He visited Germany early this year in May and was able to have a visit on our office and on our lab. He is an expert in cyanobacteria, and also the strain we were using was provided by him. Last but not least, he is also the builder or the creator for the M2 media we used for our co-culturing experiments. He advised us how to use it. He gave us the protocols for that, and we were able to build our experiments with his knowledge. Collaboration, yes. First, for our model, we had a huge supporter. This was by iGEM Tech de Monterey. They had nice ideas, and we tried to include them into our model system. USP Brazil, they were working also with the chrome sensing records, and we sent them off chrome sensing plasmids, and they are able or were able to test them and characterize them and send us the data back. This was great. Then, Team Manchester. They worked this year with an optimization tool for promoters. We sent them the promoters we were using. They created the optimizations and gave us also back the created sequences from them. And iGEM Waterloo was creating this year an information video for the society. Information regarding synthetic biology and co-culturing. And since we were working with this topic, we were able to collaborate and have some nice ideas for this video as well. You can see them on the wiki. We have a lot more collaborations on the wiki listed. For human practices, we had several different things. We were at the March for Science, had a lot of fun. We talked to experts and invited them to our university and to our lab. Here we talked to an expert in during probiotics and stuff, since we were also wanted to produce a co-culture system which is able to be used for the research in the gut microbiome. Next, we had workshops. This one were at the Munich meetup this year. And we had a visitor, which is commonly known in Germany. This is Mark Benecke, a scientist from Germany. He visited our lab and provided and helped us with huge support. For the engagement outside, we were mostly interested in education because we wanted to spread the knowledge around. There we had presentations, and we had a lecture series in our university, which was organized by our iGEM team this year. We had outreach work, visited panel discussions, for example, talked about metabolic engineering and stuff like that. Here, we visited refugees to a university and talked about biology, synthetic biology, and we also were able to make some little experiments with it, something like extracting DNA. And now this journey comes to an end, right? This is the team. This team is not just Karina and me, they are sitting here in front, and we both want to thank our team for all the work you did this year. Thank you for being this nice team this year, and also for reaching the MEDIC criteria. This was also during your support in the audience. Lastly, we want to say also thank you. Thank you in the audience that you came along here to visit our iGEM Germany 2018. Thank you.
Thank you for a great presentation. Come on up on stage.
Great, thank you very much. That was really, really interesting, very enlightening too. I've got a question regarding your co-culture. If you had taken your wild-type bacteria and your Cerevisiae and cultured them together, just starting off with the same, I don't know, cell-cell concentration or something like that, where would the equilibrium have ended up being?
Well, we have done many cross-group experiments before we started with the real experiments, and we have seen that E. coli for sure is what will bring the culture. This is why we came up with our first system and tried to regulate first the fastest growth organism into culture.
Brilliant, thanks very much.
Thanks, good morning, and great presentation. I just have one question, and it's about Elaboratus. What's the real importance, of this organism, since what it's producing is sucrose? If you compare only the E. coli and Saccharomyces with regular sucrose against these three organisms, because usually this kind of technology is used in industrial systems, and you can have regular sucrose from almost any different raw material from agriculture.
So the sucrose production by S-Energetics makes our system more autonomous. So therefore we have not to add sugar in the co-culture as well, and the sucrose, which is needed normally to co-cultivate other organisms, can be used in other types of for the industry. For example, it could be used for food production instead of the feeding of our organisms as well.
I have a brief question for the step 2 part of it. Did you ever also run negative controls with your samples to show that actually if you weren't co-culturing because you were missing, let's say, the leucine or the lysine, depending on which one you cultured, they would actually not grow?
So when we added no lysine in our intermedia, we indeed noticed that our yeast was not growing so badly, because the yeast strain we used was concentrated for 4 amino acids, and we only added in our media 3 of the amino acids needed, not the lysine part done by E. coli, and there we noticed that these organisms were dying in our culture.
I've got another question here. Sorry, it's a bit of a weird one maybe. Are cyanobacteria plants?
negative bacteria to be more exactly. So they're not eukaryotes, they're prokaryotes.
Thank you.
Thanks for a nice presentation. You mentioned that the Synagococcus species grows slowly. Do you know if it grew more slowly or faster in your culture than it does normally?
Sorry. We did vigorous experiments as mentioned previously, and it seems that growth with other organisms does not impact its growth, so growth remains stable and it's still slow.
Okay, thanks.
Hello guys, I have a question about the application. Is this system is going to be limited to the 3 strains or 3 bacteria that you have, or are you planning to modify different bacteria and try to use this same system?
Yeah, so the idea was constructing a modular toolbox that can be applied to any organisms, but we chose these 3 as our model organisms because they're well established in the lab and the literature, so it was easier to work with them than other organisms. But the idea was that eventually we can apply the systems to any organism.
Okay, thanks again to the team of Duesseldorf. Really great job. And
Mache ich das jetzt aus?
Ich muss einfach gehen.
In die Kamera gucken. In die Kamera gucken. Ich weiß nicht, wie man das ausmacht, deswegen lasse ich es laufen.
Was ist los?
Ich weiß nicht, wie man das ausmacht, deswegen lasse ich es laufen. Kette. Ich will noch einmal feststellen. Der Boy, den dauert an. Wenn sie guckt, immer wer das mal so macht.
Ja, das